see. Testing, testing. 1, 2, testing. Testing. Okay, that's good. Thank you. All right.
Okay, cool. You should be all set.
Lovely, lovely introduction. It's really a pleasure to be here. I've had such a great day already chatting about chemistry, malaria, conservation, infection, ecology. These are all things that I love, and it's great to meet you all and see such a great room of plant fans here, interested in plant taxonomy as well. So I'm going to be speaking today about the research program that I lead at Emory University, where we are looking to medicinal plants in a new way to better understand their potential for applications in drug discovery and in innovations for therapies to address the emerging issues of the post-antibiotic era. If any of you are on social media like Twitter or Instagram, I'm fine with you taking photos or tweeting at the talk. This is my Twitter handle if you want to tag me. Okay, so I'm a bit of a walker around. So to start with, I think it's important to frame the situation. We know that on Earth we have anywhere from 370 to 380,000 species of plants. And roughly 7% of those, or around 28,000, have been documented for use in traditional medicine for some sort of application. These are just a few examples of some of the more common ones that you might be familiar with. Plants like willow and the source of aspirin, foxglove for heart disease, mayapple for the treatment of warts and also for cancer, and poppy, of course, as a source of pain therapeutics. But if you look at the overall number of plants that have this long history of use in traditional medicine, the lens of Western science has only been applied in an in-depth way to a few hundred of those. So if you're looking for a field where you don't have to worry about getting scooped, this is it. There are literally thousands of species that really require dedicated scientists to get in and explore their dynamic chemistry and bioactivity. I like to include this slide in all of my lectures. Now, this is not an organic chemistry test, but I put, the cheat sheet names of each of these. And what these all hold in common are two core things. Number one, these are all compounds that are included in the World Health Organization's list of essential medicines. These are considered medicines that are core to any medical facility. And number two is that they're all from plant compounds that are derived originally from plants or are modeled after compounds derived from plants. And these have been used for a number of different applications, ranging from cancer therapies to the treatment of infection, heart disease, and pain, and the list goes on and on. So I like to include this in the beginning of my lectures to remind especially the core medical community that while we have many drugs that are synthesized today, the core basis of our Western pharmacopoeia is still actually based on natural products and are further analogs of those products. In addition to their relevance to the discovery and development of new drugs to treat patients within a Western medical context, plants actually are a key and integral part of medicine across the globe. In many developing countries, plants represent the core pharmacopoeia for 70 to 95% of people. This means that in a time of a changing climate, when some of these plants are at risk due to changes in land use practices, to changes in temperature, periods of drought, and so on, that we're not only putting at risk potential food sources and supply lines of cultivated crops, but also these wild species that are important for medicine are also under threat. So I can describe myself as an ethnobotanist. An ethnobotanist is a scientist that studies the ways that people interact with plants. It's that human-nature interface. Ethnobotany can encompass everything from the study of plants used as medicine, as musical instruments, construction materials, plants for food, and so on. I tend to focus more on the medicinal and food applications. But the core of ethnobotany is really that it is the science of survival, because without human knowledge of how to use the natural world, we would not be able to survive in all these different landscapes and ecosystems in which you can find humans. Now, one of the major targets of our research group is in looking for solutions to deal with what will be known as the post-antibiotic era. I'm a big fan of medical history and scientific history, and so when I think about the history of medicine, I'm not just going back 100 years, and by the way, we've only had antibiotics for less than 100 years, but I'm going back centuries and millennia and taking in the realization that people have actually used, again, plants for millennia for the management and treatment of infectious inflammatory diseases for a very, very long time. Now, no doubt, when antibiotics were discovered, it was a game changer in medicine. It was probably the game changer of medicine. It allowed us to undertake surgeries and other interventions that would have never been possible with the survival rates that we have today. However, today, we also face this rise in resistance to antibiotics. There was a report that came out by the UK government, and in this report, they estimated that today we have around 700,000 deaths globally due to untreatable infections. And big numbers are sometimes hard to wrap your mind around, so I like to quantify these in terms of airplane loads of people. So if you've got like a jet going across the Atlantic, that's how many airplane loads of people we lose every day globally due to infections that we do not have the tools to treat. By the year 2050, this is estimated to be this many airplane loads of people per day, or actually per hour, or roughly 10 million per year. A recent report by the CDC just showed that 35,000 deaths are currently occurring in the US alone from antibiotic-resistant infections. So we have a major problem. Now, when I give these talks that are aimed more towards student seminars, I like to integrate a bit of a personal story because for me, science is part of my life. It is my personal story. The making of a scientist really begins from your childhood experiences. And so I'm going to introduce you to my parents. This is my mom and dad. And that's me. I'm the little cutie in the middle. And you may notice that on one of my legs, there's a cast. See, I was born with multiple congenital birth defects of my skeletal system, which meant that I was missing bones in my right leg. My fibia and my femur were, my tibia and femur were very short. My fibula on my right side was missing. I had ankle bones missing. And so as I grew, you could see there was this increasing limb disparity. And so at the age of 3, my parents were faced with the choice of what to do. And at the time, the recommendation was an elective amputation, because had they not amputated, I would have to, wear really high heels for a very long time and also face the risk of chronically broken ankle because of those missing bones. Now, unfortunately, after that surgery, like many other people have faced in the hospital, I acquired a nosocomial infection. That means a hospital-acquired infection. I was sent home, unbeknownst to the doctors, that I had this infection, and after a few days, my mom smelled something was off. Just smelled wrong. There was this cloying, sweet scent to my stump that smelled really wrong. And so against the doctor's and nurse's advice, she unwrapped the bandages at home. And as she did, a pool of pus and flesh came out like Jell-O. Sorry for all of you that are about to eat lunch. And my bone was exposed. I had gangrene and a very necrotic staph infection that had started to enter the bone in a process known as osteomyelitis. This is a biofilm infection of the bone. And so I had to go back to the hospital. They debrided the tissue, and as my mother describes it, it's something I hope I never have to experience with my kids because they had to scrape out all the dead tissue while I was still awake, put me into betadine baths, which I thought were blood baths. I literally thought they were putting me in blood. And I had a little rubber ducky in my blood bath. It was very traumatic. Later, they clipped the bone back, and I'm now able to walk. They're able to save some part below my knee, but I have a much shorter residual limb than most amputees. So you're not going to see me doing these super athletic events with a super bouncy leg because I have basically no tissue under my stump. So the point of this story is to tell you that even in the period of the 1980s, when methicillin-resistant staph was not really out in the clinics, I still almost died from a drug-sensitive strain. Imagine if this had been a drug-resistant strain. And one of the reasons it was so difficult to treat was also because it had entered my tissues. It had entered the bone. And that's what required cutting back the bone. So as I go and talk about more of my research on biofilm, remember this story. Now, what are the challenges that we face today in drug discovery? Well, the history of antibiotic discovery is really fascinating. There were major players. There were times in which we were able to scoop up soil from across the globe, and using simple techniques that were brought forward by Watson and colleagues, that we were able to then cultivate interesting soil microbes and isolate bioactive compounds. But after some time, we entered a period known as the drug discovery void. Now, a couple of things happened during this period that are really critical to the history and the future of antibiotic drug discovery. Number one is we had basically harvested all the low-hanging fruit, all the easy-to-cultivate organisms, and we weren't finding new chemical scaffolds. This also coincides with a time when we had great advances in robotics. So you had a shift towards high-throughput screening enterprises that allowed us to test millions of compounds very rapidly. It is also a time when we had advancements in the field of combinatorial chemistry, which basically allows scientists to create large chemical libraries of man-made molecules. And so the thought process at the time was, if we can create millions of these molecules in the lab, push them through our robotic systems, that surely we're going to find lots of really important and interesting drugs, and this whole natural products business, which is very difficult to undertake, we're going to put to the side. So you had a decline in the emphasis in natural products, an increase in this CombiChem high throughput approach. Now, who can tell me how many antibiotics came out of this investment? Zero. Zero. Big fat loss, right? Now, it's easy to be 20 years, 30 years in the future and say, well, that was a dumb idea, right? But some of the best ideas sometimes don't always play out. And so one of the consequences of this, of course, was also this loss in momentum with some of the momentum that we had experienced in natural products. And so as you can imagine, I'm a big supporter of natural products research, because I think that the relevance of these molecules that are already being used in biological systems through different ecological signaling pathways between organisms is a ripe area for picking new bioactive molecules. So how do we go about finding these molecules in my lab? Well, as an ethnobotanist, we focus on data sets of knowledge regarding plants that people use today or used in the past as medicine. So we look to historic texts for pieces of evidence, and we also look to current day practices with plants. What I want to make very clear here is that this is not a random approach. It is the exact opposite of a random approach, because we are only focusing on those plants that people are using for the treatment of infectious and inflammatory disease. We are only working on those tissues of those plants that people are using for these diseases. So this is not a random grab soil here from there and see if something works. This is very targeted. We're trying to understand how and why and which molecules are responsible for this history of use. It involves a number of different disciplines. We have to involve botany, so we have botanical identification, deposit in our herbarium of our specimens. Authentication of the identity of these plants is incredibly important. We prepare extracts. We then undertake bioassays to explore the potential bioactivities of these molecules. We currently use a classic system of bioassay-guided fractionation. My hope is that with advances in metabolomics, advances in new emerging technologies like cryo-EM, micro-ED, that we might be able to do less of this kind of isolation-based approach and be able to look more and more at these complex mixtures. But for now, we're using this classic approach. We go to active compound identification, and then we work with our friends that are medicinal chemists that can then generate analogs and undertake some of these structure-activity relationship studies. And our goal is to get all the way down to drugs. Now, where do we collect? This is a map of the world's terrestrial hotspots of biodiversity. And again, our intent and our direction in collecting is very much targeted. We are focusing in areas in yellow here that are highlighting regions of biodiversity. I do most of the collecting personally in the Southeast US and also in the Mediterranean region. We work with our partners through collaborations in South Africa, in Pakistan, China, Uganda, Colombia, and so on, in collaborative activities. I wanted to also highlight the importance and value of the herbarium. If you've not been to the Massey Herbarium yet, I highly encourage you to go give it a visit. It's a beautiful collection. They have over 115,000 specimens of plants, and these natural history collections are really important to this kind of drug discovery because, again, they serve as that key in authentication of these sources of compounds. And so another important factor to consider when you're doing this type of ethnobotanical research is to ensure that ethics are a big part of your fundamental approach from start to finish. So there are international conventions, including the Convention on Biological Diversity and the Nagoya Protocol, that establish guidelines for research within this space. So one of the perspectives that I take personally, and I've been doing this since I was a graduate student, is to start benefit sharing from day one. So one way that you can benefit share, again, even if you don't have millions in revenue coming from some future potential drug, which, by the way, may not ever happen, one way to begin is by returning the knowledge that you've documented back to the communities in a way that's accessible to them. We've done this through construction of ethnobotanical gardens. We've done this by writing books and pamphlets in their local language. We've done it through workshops. And now we also do it, now that I'm a PI, I'm able to take in students from different countries, especially in these countries where we're working, give them training opportunities that they're then bringing back to their countries. We're also engaged in a number of university capacity building projects sponsored by the State Department in the Balkans. So we're also helping build up research infrastructure at universities like the University of Pristina in Kosovo. I wanted to show you some photos of some of the different areas where we collect, and again, our collection sites are intentionally ecologically quite different, and they're also based on work with different ethnic groups. So this is some of our work that we do in Albania and Kosovo. We work along the Shari Mountain region, which is also where the Balkan conflict was fought in the 1990s. So these are people that are very isolated. Roads are very difficult to traverse to these villages, and as a result, they have an incredibly rich sense of their wild flora that they use for food and for medicine. I also work in islands around the Mediterranean. I show this video just to also say to anyone that thinks about wanting to have a family while you do science, it is totally possible. I have 3 kids. They come with me in the field. If we have to go up the mountain, I just get a couple extra mules, and there we go. And so you can see them strapped on with the plant specimens hanging off, and the fit graduate students, of course, have to walk. But and these island environments are really amazing to work in, again, because you have populations of isolated people that are also engaging in trade across islands. They're trading. This island has more species suitable for firewood. They trade with another island to get access to their bread. They trade with another island to get access to certain fruits and vegetables. And so there's these very interesting dynamic networks involved here. In the U.S., we work in some really interesting sites in the southeastern U.S., both in oak hammocks and swamps of southwest Florida, where I actually grew up, and then also in South Georgia. We're really fortunate to be able to collaborate with the Jones Ecological Research Station at Itchaway, and this is actually a 30,000-acre longleaf pine forest habitat that was once owned by the former CEO of Coca-Cola, Robert Woodruff. And so it's great for us being based in Atlanta because it's only a 4-hour drive, and I can bring a lot of my students, including undergrads, down there for research and training. Now, one other thing that is difficult that I wanted to highlight here is that field work as a mom that has one leg presents its challenges. And so I like to put this slide up just to remind any of the students in the room, we all face our own challenges. Right? We all have different challenges, whether they be physical, mental, spiritual, economic, social, but you can accomplish what you go after it with proper planning and with the right support. So I like to put that out there. But also I want to show you some reality. This is my Instagram-worthy morning photo. I'm very excited when I'm in the field, and this is the reality of when I get home. So you can do a lot, but expect to work really hard, okay? So what do we do in the lab? We have field research, we have the herbarium, we have the phytochemistry lab, and the microbiology lab. So these are the 4 domains where I lead my research group. In our library, we've constructed, again, this very specialized library of plants used for infectious and inflammatory disease from across the globe. We have 600 species that we've collected and over 1,900 extracts and growing. What's unique, again, about this library is that it is targeted towards ethnobotanical use for human medicine and for food. So what are the organisms that we're targeting? This is a recent report that came out, again, by CDC that's kind of categorized these different microorganisms by their threat level. And this is an effort to encourage scientists to focus on some of these. And so the ones that are underlined are ones that we work on heavily in my lab, plus there are additional ones. We also work on organisms like acne. Again, I'm very interested in any kind of skin pathogens. So I'm going to give you some examples, just some highlights of some of the different projects that we have ongoing in my lab. I'm going to start with Staphylococcus aureus, my old nemesis from childhood. And I just want to highlight the point that staph, although we do have a number of drugs that in a test tube work very well against staph, this is still a major killer of patients in the US, from complicated skin and soft tissue infections that can also devolve into diseases like sepsis, where you have multi-organ failure as a result of these types of infections and bacteremia. Now, staphylococcal toxins are responsible for a number of different diseases. This can range from toxic shock syndrome from the use of superabsorbent tampons to scalded skin syndrome in neonates, where the baby's skin basically peels off. You have the hallmark, which is really abscesses, necrosis, and sepsis, and this is due to these hemolytic or blood lysine toxins. And then we have atopic dermatitis, in which we know that staph plays a very important role in those atopic lesions, and we know that certain toxins help to drive disease. So one of the approaches that I got interested in early on in graduate school was this idea of quorum quenching, or interfering with bacterial communication pathways. Because as I studied more and more plants, I found that not all of them actually inhibit growth or kill bacteria. So they didn't all work like you would expect an antibiotic to work. But yet, as I talked to people in the field, they were saying, no, we use this for infection and it works. I have this boil, I treat it with this, and the boil is healed. So how is it working? That was the question. So the idea of looking at virulence inhibition or through interfering with quorum sensing is one of those questions I started to ask. So in Staphylococcus aureus, the accessory gene regulator, or AGR, is the major control system for virulence in Staph aureus. I won't go into too much detail on this, just to let you know that you have these AIPs, or auto-inducing peptides, that are released into the extracellular environment. When you reach a quorum, or a certain density of these signals, it upregulates events that lead to greater production of these secreted virulence factors. Now, in the lab, we can use tools ranging from our kind of fluorescent gene reporter strains to also tools like HPLC to track the production of these specific toxins. We also have noted that not all staph behave the same. We've looked at large libraries of different strains of Staphylococcus aureus and have noted that very clearly different strains have higher capacities to produce certain toxins over than others, even though they have the same genetic framework to produce these toxins. Now I want to get to the first example of a historic text that helped lead us to the discovery of a quorum sensing inhibitor. This is an example of a text. This is from Historia Naturalis Brasiliae that goes back to the 1600s. It was written by a Dutch naturalist, William Piso, in Latin. And this, as well as other texts that came later in the 1800s, highlight the applications of a plant known as the Brazilian pepper tree, it's also called the Christmas berry tree, for the treatment of infections. I got really interested in this top application as a balm for wounds and ulcers, and you can see it has some other kind of infectious disease applications. And so what we did is we collected this plant. By the way, this plant is also a noxious weed in the state of Florida. It's probably the most hated plant in Florida where I grew up. It's abundant. Everyone's happy to give me as much of it as I want. But again, it has this long history of traditional medicine use in Brazil, where it still is part of the Brazilian pharmacopoeia. So we wanted to find out what is responsible for some of this activity. This is just showing some of the next steps of going through iterative steps of bioassay-guided fractionation to get down to some of these compounds. The first thing that we did is we looked at our fluorescent reporters for AGR, that accessory gene regulator system. There are 4 types of AGR based on differential structures of those autoinducing peptides. And what we were able to do is track the growth inhibitory properties of these versus the quorum sensing inhibitory activities. So you see that these compounds are not actually inhibiting growth, but they are inhibiting their ability to signal with one another. So if you look at the actual tube of bacteria growing, it's turbid, it's dense in growth, but they just can't sense each other. It's like they're tricked into believing they're alone. And when bacteria think that they're alone, I'm using think as a, they're not really thinking, but when they don't sense each other, they behave differently, because they behave differently when they have this kind of group dynamic. And so in the next step that we asked is like, well, how does this work in our murine models of infection? In this model, we take a hypertoxin producing strain known as the Los Angeles clone, inject it into the dermis of mice, and then co-inject it with our compounds. And what we found with this is with 430-DF5, which is a enriched fraction of the pepper tree extract, we found very clearly that you can see a decrease in necrotic lesion formation. So it forms a much smaller lesion than the untreated form in the mouse. And what that means, again, is we're not killing the bacteria, we're just knocking out their ability to create these toxins. Now, one of the challenges here, of course, is that plant tissues are very complex, even in our refined.
Can I ask you a quick question?
Yeah.
About the previous slide? So this is one dose treatment?
Yes.
So what happened after the effect of the drug, of your potential drug?
For this one, it was only tracked to 7 days.
So does it come back?
I don't know, because it was only, they were sacrificed at 7 days.
Oh.
So it's a proof of concept.
Oh, I see.
Yeah. Okay, but that's a good question, because that's one of our next questions we want to ask is, can this be effective in treating an already established infection? This is a prophylactic model that's a proof of concept assay. Okay, so the next question is, we saw proof of concept in a mouse that there's some activity, but what's responsible for that activity? And you can use tools like mass spectrometry to get a good idea on the complexity of molecules that are present based on accurate mass measurements. However, there's a lot that's not known. There are a lot of unreported compounds, especially when it comes to plant natural products. So this is at best a good estimate or guess. And so one of the hurdles we ran into in the lab was the ability to isolate enough of those individual peaks. And this is where I really am excited that we have such an innovative and creative group, because I came into the lab one day and I saw one of my postdocs just watching the clock and taking manual fractions because we didn't have a large-scale fraction collector. And I said, surely this is not a good way to spend your time, and you must be going crazy watching the timer as you do this. And so we asked in the group, we started talking about this, and a member of the group said, I think I can make something. I'm like, okay. And he's like, well, I'm going to go get some Legos. I'm going to make something. I'm like, okay. And so this is what he actually did. He took some materials from a hardware store. This is just from a local hardware store of some plywood and some C-channel metal. The Lego Mindstorm control unit, which we use as our computing unit because it has free programming software available online, and then a bunch of different Lego pieces. And so actually, this enabled us now to do preparative HPLC basically around the clock. We can now run 10, 11 runs of prep into these larger scale flasks to get enough compound to now have even crystal structures of some of our natural products, which is really exciting because when I was trained as a student, you just didn't see many crystal structures of these natural products because it's hard to get to isolate enough of them because some of the active compounds are only present, at less than 0.01% of your total extract. And so as this was happening, one of my students, this is a visiting student from China who did some fantastic work. His name is Huachao Tong. And he came to me and he was very upset. He said, Dr. Quabe, look, I can't get this to go back in solution. And I said, what? It had crystallized on its own. So I was like, take it down to the X-ray facility. And we were actually able to get crystal structures on each of the bioactive molecules that we were able to isolate from pepper tree. So that was like a huge leap in our lab that had not been previously possible, and it was all thanks to this Lego platform that cost less than $500. Okay, so now we have these 3 individual compounds. The next question again is go back to proof of concept. They behaved quite similarly in the bioassays. We wanted to see how do they behave in vivo. And what we found here is there were some differences in the activity. Compound 2 had much greater effect than compounds 1 or 3, but all showed benefits over the vehicle control. So right now we're at the stage where I'm trying to get funded to go on to do much more intensive in vivo tests to look at this not from a prophylactic model, but also from a therapeutic perspective. For example, can we rescue mice from bacteremia? Can we use this effectively as a therapy for atopic dermatitis? There are a lot of disease questions that we want to ask. We just need to get study section to agree with us. All right. We also did something very similar in our work with the European chestnut. I'm not going to go into that whole story, but just to note that, again, we were able to isolate crystal structures of these really interesting hydroperoxy cycloartanes that do have some light reactivity, but we found that one of these was quite stable and is showing some interesting activity. We're still waiting on the mouse data to come back on that. Okay. The next bug I want to talk about is carbapenem-resistant Acinetobacter. So this is a gram-negative pathogen, unlike staph, which is gram-positive, and this is a major source of, it's a major antibiotic-resistant pathogen that's incredibly difficult to treat. And we found it was incredibly difficult to treat. We screened our entire library and only found a few potential hits. Out of those, we were able to isolate a compound that just shows some promising activity. Unfortunately, I'm still not sure if it's going to be enough to really advance forward because the MIC is in the around 64. So it may not be a good enough drug, but it could be useful as a tool compound. We know that the therapeutic index, when you compare it to human cell toxicity, is pretty good, but again, it may be a good candidate also for MedChem to play with. I can't put the structure up yet because we're still waiting on some patent stuff. What was interesting, though, with this particular compound that really I found fascinating is that when we compared it, when we looked at its capacity for resisting or for generating resistance, because we were trying to isolate mutants of this to see if we could figure out what is the molecular target or the mechanism by which this particular drug works. And so the way this experiment works is you basically grow the microbes in the presence of your drug and basically take colonies from each generation. You do this for a series of 21 days. And what we found in comparing this to tetracycline, here you have tetracycline, the darker bars show how the dose response curve continues to move to the right, meaning that this is becoming more and more resistant. But if you look at the dose response curves in SF4, which is our compound, they stayed tight. We weren't able to generate resistance, which was really frustrating when you're trying to get a mutant, but really exciting because it's showing that it's not easily acquiring resistance. Now, another organism that we're interested in that's also we looked at SF4's activity against is Candida auris. Candida auris is a fungal pathogen that has emerged on the scene as a major threat. Up to 60% of patients that are infected with this, which are usually immune-compromised patients, die from these infections. And this is one that people are keeping a really close eye on. There are a lot of emerging fungal threats because, in large parts, to our excessive use of antifungals in our agricultural systems, including, roses. I kind of hate it when my husband brings me roses now because I'm like, how many anti-fungicides have been sprayed on these and what kind of microbes are present? But this is one that's interesting to study. So again, we looked at this same source, 429, found that SF4, interestingly, was very active against these fungal pathogens, including not just Candida albicans, but also these multi-drug resistant strains of Candida auris. We only have basically 3 functional classes of antifungals. Candida auris is usually resistant to at least one of those, sometimes all 3. So we do need new drugs for this. And here we had some very nice MICs, less than 1 microgram per mil. So this is another area that we're trying to push forward on. Now, I promised to give some examples of some other early texts that we looked at that were important for validation of traditional medicine. I usually don't work on plants that are really common in the marketplace, plants like ginkgo or St. John's wort, and I ended up getting interested in ginkgo because of something one of my undergraduate students found. She's originally from mainland China, and she was working in, I sent her to the library at Emory and told her to look at some of the old texts that we had. And she started getting interested in this text, this is the Ben Kao Gang Mu, and what she found was that when it came to ginkgo, it wasn't so much the leaves that were being used medicinally historically in traditional Chinese medicine, instead it was the seeds. And there was a unique use for the treatment of infections that hadn't really been explored before in the literature. And so we took the seed kernels and looked at their activities. These are some of the examples of how they were used. They were used raw, pestled raw and applied to genital ulcers, used to treat dog bite abscesses, used to treat mastitis. All of these could have obviously a bacterial implication with these types of skin and soft tissue infections. Bullae, pustules, furuncles. I get really excited about oozing pussy wounds. And so we looked at these in the lab, and indeed we found that there was some activity especially when it came to biofilm inhibition. It wasn't like super stellar, but there was some interesting activity that could have validated the activity of these. One of the problems, there was also some growth inhibitory activity. One of the problems, though, was that this also exhibited a good deal of cytotoxicity due to some of these compounds. So not necessarily a great therapeutic candidate, but it did give us some insight into how this may have worked historically. Another area of historic medicine that we looked at, and this was with another undergraduate student, Micah Detweiler, he was interested in plants that grew around our campus. And so I said, why don't you go back to look in medical history of the Civil War conflict? Because we knew that during the war, infectious disease was a major issue, especially diarrhea, dysentery. And if you've read the medical records from the period, which I have, there were a lot of STDs, like a lot in these camps. So they had a lot of issues with infectious disease. And one of the things that was interesting, and this is where you can kind of ask a broader question, is what happens when your source of medicines that you're used to of that era are cut off? So the Union blockade actually prevented the importing of medicines like morphine, of a lot of these emetics and other pharmaceuticals that would have been used to purge the body of disease at that time. And so what did they do? Well, the Confederate Surgeon General actually commissioned this guy, Dr. Francis Porcher, who was both a botanist and physician. Remember, in the past, botanists were often, or physicians often had a botanical background. And he wrote this book called Resources of the Southern Field and Forest, which was based on, of course, Native American knowledge and also the knowledge of enslaved Africans in the South. This was then used to create a medic supply table that was then used to stock the field clinics for treatment of these soldiers. And so we looked in this list and we decided to focus on just 3 species. There were many others still to study, but one that you might all recognize from the emblem of the Massey herbarium is, of course, the tulip poplar, or Leodendron tulipifera, white oak, and then devil's walking stick. And so Micah went onto campus and collected these. And again, what we found is that there were some modest activities in blocking quorum sensing and also in blocking biofilm inhibition or biofilm formation in a number of different pathogens. And so I'm going to wrap up with one last example, and this is where I'm going to get a little bit more into what biofilms do. And when you wake up in the morning and you rub your tongue across your teeth, anyone do that? You feel like kind of slimy, gritty feel in your tongue? That's this. This is a biofilm, because I've been talking about it very abstractly, but you can see pictures. This is what it looks like. They're often polymicrobial. They're often enter this very slow growth, slow metabolism phase. Now, antibiotics work best against rapidly dividing cells, so that's one way that they evade activity of antibiotics. And also this kind of slimy, goopy polysaccharide matrix they produce also serves as a physical barrier to therapy and to your host immune response. So one of the early studies I worked on was in the documentation of plants used in southern Italy for the treatment of boils and ulcers and, again, these oozing infections. And one that I got really interested in was the use of the elm leaf blackberry, Rubus elmifolius. And this is one where they would actually take the leaves, combine it with some pork fat, and apply it topically. Now, the pork fat could actually play an important role in helping movement of compounds across the skin barrier. And so in the lab, we knew that in the presence of extracts from this plant, this is a confocal laser scan microscopy image. The black shows no attachment, the green shows attachment, that you had this dose-response activity where, again, without inhibiting growth, you were able to block attachment to surfaces. And so one of the questions became, well, okay, we can stop things from sticking to surfaces, which is really important for medical device infections, but what do we do if a patient already has a medical device infection? So when I talk about medical devices, that means temporary things like IV catheters, urinary catheters. It also can be something more permanent, like a hip replacement or a knee replacement or metal pins in your body. People, as long as you have those artificial devices in your body, you will be susceptible for these kinds of infections the rest of your life. All it takes is a scratch of your nose, bacteria to survive, passes through your bloodstream, and they will land on those devices. When you do have an infection in one of those devices, it usually requires surgery, debridement, again, scraping out that tissue, intensive antibiotic therapy, removal of the device, put in a new device, and then in some cases, some patients have recurrence rates of up to 40%. So you can see why people that have chronically reinfected knee replacements elect for amputation, because going through that process of infection is just horrific, and then they end up with C. diff. There's like a whole cascade effect. So we need better therapies that can attack and remove these biofilms. So our question at first was, well, can this extract that blocks biofilm attachment be useful in removing an already established biofilm? And so in this study, we took segments of IV catheter and basically just threw it into media with the bug. And we basically would rinse them each day and then treat them each day. The first answer, of course, was that you can see the extract 220-DF2 and our control, there was no difference. So this does not work in blocking or removing it once it's already formed. This is 10 times the dose of antibiotic, and even after 6 or 7 days, there's still no real efficacy in removing those adherent cells. But what we did find is that after 6 days of treatment, 5 days of treatment, when you combine the antibiotic with this biofilm inhibitor, we did start to see reductions in numbers of adherent cells. So that was exciting. So our next question was, of course, well, what is it? Going back to that complexity issue, we knew that we had mixtures of ellagic acid glycosides based on our mass spectrometry analyses, but they were really difficult to separate. And every time I separated them, I lost activity. So one of the questions was, well, could this be due to synergy? And so in order to do this, we actually worked with one of my good colleagues and friends, Emily Weinert, who's now at Penn State. She was at Emory before. And I asked her, can you synthesize a variety of different ellagic acid glycosides for me so we can test them individually and in combination? And so she was able to do that. We recombined the parts, we tested them, and what we found was, well, ellagic acid rhamnoside, which is a specific kind of sugar unit that's attached to the ellagic acid, seemed to have the best activity. None of them were as good as the actual plant extract. So now we say, well, where do we go with this, right? So the other options are you can pursue this FDA botanical drug pathway, which allows for drugs that have complex mixtures of compounds, or you can go another route. So this is the route we're currently trying, and this is the medical device route. So this was licensed out to my company Phytotech, which is now in partnership with Alira Health to create this eco-friendly bandage known as PureMend. We're still in the prototype development phase, but basically the idea is to combine the biofilm inhibiting extract with other plant essential oils that show polymicrobial antimicrobial activities, and then develop this as a bandage to treat patients that have chronic non-healing wounds like diabetic foot ulcers, venous leg ulcers. In some cases, these patients can have these open wounds for months, and they're very foul smelling and very difficult to care for. And so we're hoping this might offer some unique opportunities to use these botanicals in a way to help these patients. So what are some of the ways that we collaborate with folks? We, of course, can collaborate on the side of ethnobotany. We can work with folks on botanical authentication. But our main bread and butter is also in this, on the lab side, within chemical characterization of these complex mixtures, and also large suites of antimicrobial and pharmacological tests that we can run on natural products. And so for all the students in the room, and I'll even give it to the professors, this is your quiz. So let's see if you were paying attention. How many of you can answer this question? Is there a role for botanicals to play in the search for new solutions to AMR? Yes, no? Yes. All right. Yes. Pass. Good. Will all effective botanicals for infection control act in the same way as standard antibiotics? Yes or no? No. Yes. You're almost there. Okay. Last question. Can bioprospecting be done in an ethical way that ensures access and benefit sharing? Yes or no? Yes. Okay. You guys pass. Excellent. I want to thank my collaborators, my funders, my amazing team. You know, I always say I'm the dog and pony show, but they're the ones that are actually really plodding away in the lab. And it's so exciting. It's such a great experience to be able to be a professor and work with so many energetic young people that are just excited to do science. And I wouldn't give it up for anything. I also am going to have a shameless plug for the food podcast. If anyone is interested in how food and health and ecology and chemistry and culture are intertwined, give it a listen, leave it a rating. I'd appreciate it. Thank you. We have questions. Yes. Mm-hmm.
And my partner is in Chinese medicine and so practices herbalism practice.
Yeah.
And one of the things that I see often not talked about a lot is like who ends up using this knowledge in the form of patents and who ends up profiting through the patents.
Mm-hmm.
Whether there are other modes of making these, for example, antimicrobial herbalism products available through the traditional knowledge practitioners and that are already practicing, rather than through, you know, a medical system that really approaches diagnosis and the whole approach to creation of knowledge around health in a completely different way.
Yeah, so there's a number of questions in there. I'll try and tackle each of them. Number one is, as I said in the beginning, one of our big initiatives is on the ground, is like we not only return knowledge that we're documenting back to communities, but we really try and communicate back our laboratory findings back to those communities. In fact, I had a Fulbright fellow from Germany who's doing the work in Uganda, and he asked the healers he was working with, what do you want most from this study? And unanimously, they said, we want to know how these work in your laboratory studies. We want you to come back and explain this to us so we can better practice our medicine. And that's one way of many that we bring back knowledge and help to reinforce and, again, build capacity for research at the community and university levels. The second question on patents. The ways that this works is that you have agreements with universities in these different countries. You have agreements within communities. The patent law follows standard legal practice of the innovation that comes out of discovery of molecules and utility of these molecules for medicine. The reality is that in order for medical innovations to reach the broader world, which is what my aim is, to make medicine better for everyone, you have to have protection of that information in order to get it developed down the road. Now, you can do this in an equitable way that ensures that revenue streams go back to those sources, those knowledge holders. And in fact, that's what the Nagoya Protocol is all about. And there's lots of case studies you can read into there on their website if you're interested in that. So I think that we have this kind of mentality of pharma is bad sometimes, but I think that if your goal is to make medicine available for everybody, you have to work within those economic models to make it available on a broad scale. So that's my perspective on it. Any other questions? Yep.
Yeah, I was interested in the biofilm work, and you mentioned how the results were best when you combined the compounds and the antibiotics. So when you're thinking about antibiotic resistance, is it generally thought that the microbes are resistant to the antibiotics, or because they're forming biofilms, they're resistant to the antibiotics? I just don't know enough about the field.
That's a great question. So there's really two divisions of antibiotic resistance, or two different major pathways. You have acquired resistance, where you have exchange of genetic information between different strains of the same species, or even between species, where they can exchange genes that encode for pathways to produce enzymes that, for example, degrade antibiotics, or genes for efflux pumps that pump the antibiotics back out of the cell, and so on. That's acquired resistance. Then you have intrinsic resistance, which is really about like their ability to naturally evade any kind of attack through something like a biofilm. And this is where it gets tricky. And again, this is what we see in staph, like the differences in the clinic. If I take the bacteria that I isolate from a patient that has a staph infection and I put it into a test tube, staph is actually pretty easy to kill in a test tube, but not always easy to kill when it's in the patient. And this is because of these other intrinsic modalities by which they can evade activity.
Do we know if they can acquire intrinsic resistance? Like, can bacteria become?
So there are some strains that tend to be better producers of biofilms. I don't know enough about the microbiology behind that, if that's something that's acquired or could be acquired. I don't know if anybody knows that right now. I do know that more and more studies are showing, especially with patients who have recurrent infections. I work with a OBGYN surgical fellow in my lab, and he treats women that have these really horrific recurrent urinary tract infections. They're just impossible to deal with. And so he's actually asking that question right now. Are those bacteria in those urinary tract infection patients, are they better biofilm formers? And is that part of the reason why he's having a hard time clearing these women of their infections? So intrinsic pathways are really important to recurrence and recalcitrance. Other questions? Yep.
So the whole body of work is very fascinating, and the need for testing traditional medicines, I think, is often challenged by limited funding. And as you described, if you're just looking at does it kill the bacteria, you could get a very easy answer, no, and then
it gets dismissed. Yeah.
And so I'm wondering if, not knowing anything myself about these Quorum signaling, is that, it seems like there could be a number of phenomena that are similar to that, and we just
Yes.
don't know.
You've hit on it.
To test that, it seems like the
Yeah.
animal models are really crucial for your mechanism to work, right? You could demonstrate that it does have the effect that the traditional healers claim,
Mm-hmm.
and that would encourage you to keep pursuing it, as opposed to saying, well, I can't even replicate the effect. Maybe that's not one with.
Yeah. I think one of the limitations in our field is that, well, let me back up. It's paradigm shifts in understanding scientific processes are often difficult for people to accept, right? I give the example to my students, of this theory of spontaneous generation, the idea that living organisms emerge from just inanimate objects. It took 300 years of experiments to prove that wrong, right? When it comes to infection, our gut response is to stick with what Fleming discovered, and you kill, kill, kill. We now know, as we understand more and more about the microbiome, how this can have a lot of other collateral damage. I think one of the most important lessons we can learn from traditional medicine practitioners is that there's more than one way to address the problem of infection, because your question shouldn't be around how well can I kill this organism. It should be more around how can I heal this patient, and are there different avenues in which to achieve that goal. There are a lot of questions that I am not asking of these plants, and I know that. Those are like major failings in my research program, in that we're not looking at the impact on immunity. We're not looking at impact on the host, because some of these plants might not be working at all on the pathogen, but they're hitting the host. They might be working on pathways and microbes that scientists have not even discovered yet. And so I think what I try and do is I try and keep an open mindset. When I test something against our models and it doesn't work, I never say, oh, well, that's just junk, that plant doesn't work. Because, I think that's like the epitome of arrogance to say, well, if it doesn't work in my hands, it doesn't work, right? Even though it's been used by people for thousands of years. I think we just haven't asked all the right questions. And so one of the things I really try and encourage my students to do is to also work where there is no box. You know how they always say, think outside the box? Like, if you're thinking in that mentality, if you're thinking outside of something that already exists, you've already lost the game. there can't be a box where you operate, you know, so you have to be just incredibly open to thinking about these different pathways. But yes, I recognize we're not asking all the questions because we just don't know all the right questions yet. Yeah.
Do you ever have to work with or need to work with endangered or threatened species, and how do you communicate with and collaborate with conservation scientists?
That's a great question. We do not collect endangered or threatened species. We work with a lot of medicinal plants are actually weeds. But think about that from an ecological perspective. What is a weed but a very effective plant at getting it? And what makes it so effective? It's chemistry, I think. It has a unique way of engaging with that environment, and that's what I think makes it so effective at surviving in these different environments. So a lot of medicinal plants, they're not, you know, you have this idea of the mythic shaman in the forest, and it's the mysterious orchid at the top of the canopy that he has to climb. No, these are weeds that are growing around their houses or in the village or near the borders of the village or near their fields. So no, we're very cognizant of that. We do work with conservationists. My most recent trip to the Balkans, I actually was accompanied by a colleague at Leiden who's an orchid conservation specialist, and she actually did an assessment of their orchid collection in Kosovo. Now, Kosovo is not CITES listed, so you can't do any kind of import or export, but what we did do is I had a Nat Geo photographer with me, and he had an $80,000 camera. He carried this thing around. I said, I want you to take the best resolution photos you can of each of these orchids in the wild and of their collection that we had pressed. And so those are some of the ways we're trying, and we gave all those images to the botanist at that herbarium so they could use them. So yeah, so we avoid collecting things that are threatened, but do whenever we can try to contribute towards conservation.
And we have time for one more question.
I'm in the very back.
Have you observed any relationship between the effectiveness of medicinal herbs with the doctrine of signatures, or is it just coincidental?
You know, that's a great question. Just to let everybody know, the doctrine of signatures is this idea that this idea around how plants that seep red latex, for example, might be good for boosting blood performance, or could be good for immunity. This idea of like looking at physical characteristics or the shape of a leaf like a liver, and it's supposed to help the liver function. There's been a lot of work done on this, and I tend to agree with the papers that look at this as more of a mnemonic device, of a way of that ethnic or indigenous people use to remember the uses of those particular activities. Because there's lots of things that look like a kidney. There's lots, but that aren't all used for traditional medicine. There's lots of latex-bearing plants that are not used for traditional medicine. So I see it more as a memorization tool that many different groups use. That's my perspective. Great. Well, thank you guys so much for your attention. I appreciate it.
If you're signed up for the student lunch, it is full, but if you're signed up, it'll be across the hall in the boardroom.
All right.
Great, that was awesome.
Thank you.
You ready to go to this reading room?